So as part of Lab 2 for 241, it was a review of cell organelles. So we have this model available for students to review. And so the obvious is that the plasma membrane is the external component of a cell. And internally with eukaryotic cells, we have a nuclear membrane that surrounds the nucleus. So the white here represents the nuclear membrane. The clear stuff represents the nuclear plasm inside the nuclear membrane. The really dark structure here represents the nucleolus, and they cleverly inserted wires into the nucleus that look like a double helix to represent the DNA or chromatin that would be found within the nucleus. So as we look at the cytoplasm, then these two structures are centrioles, and their purpose is to organize the cell division, and they exist in an area of the cell called the centrosome. And as we look in the other parts of the cytoplasm, we have these little kidney bean shaped orange things which have highly folded membranes on the inside. So these represent the mitochondria with the unfolded membranes on the inside of that, where cellular respiration occurs in terms of the Krebs cycle and electron transport system. So most of the folded stuff that we're seeing inside of the cell is dotted with these little white things, and the white things are ribosomes. So all of this folded material represents rough ER. As you'll recall, ribosomes are for protein manufacture. So rough ER produces proteins that are largely going to be exported from the cell. So associated with rough ER, there's always this apparatus called the Golgi apparatus, and it packages the proteins being produced on rough ER and puts them in little excretory vesicles so that they can be exported from the cell. Now the model has a couple of nuances. So over here where they've removed the outside with the little white dots, they're trying to represent smooth ER. So smooth ER would be the site where lipid synthesis occurs inside of cells. And then we have a number of little vacuoles, dark and light and yellow vacuoles that we can see here, which are supposed to represent things like lysosomes and peroxisomes that would be bags of enzymes that are housed within the cytoplasm of a cell as well. So make sure that you study these organelles, you can identify them, and you know what their roles are in the life of a cell. So in Lab 4, our purpose of our lab is to explore the integumentary system. And we can review some things from earlier labs on tissues while we're doing it, because the integumentary system represents one of our organs, which is comprised of 2 or more types of tissue with specialized apparatus within it. So when we're looking at the skin, we can divide the skin into some basic layers. So from the top of this folded layer right here upward, we would say is the epidermis, because it's above or outside of the dermis. And then from this folded layer here downward to the top of this areas of adipose tissue, we would call the dermis. And then the layer including all of the adipose tissue here would be called the hypodermis. So our relationship is the dermis. What's above the dermis is the epidermis, and what's below the dermis here is the hypodermis. Now the other thing we can do then is we can review tissue layers related to that. So our epidermis, starting with this row of cells right here, is comprised of keratinized stratified squamous. And so the cells are produced in this layer of cells right here, and as daughter cells are produced from mitosis, the cells are pushed upward, and they go through a progression of change till they get to these dead keratinized cells at the top, which is where the tissue gets its name, keratinized stratified squamous. And what we do in thin skin like this is we start down here with a newly formed cell, and by the time it gets to the top up here, it takes about 25 days. So we replace most of our skin in a 25-day period. Now, the epidermis itself can be divided into layers that we call strata or stratums. And the single row of cells that is a mitotic that gives rise to new cells is called the stratum bacilli. Above that, this lighter colored layer right here is where the cells are being pushed upward. They start out in a polygonal shape and they begin to flatten out. As the cells become flattened, they take on little projections. And so this layer is called the stratum spinosum. As we move up from the stratum spinosum, or a layer where the cells are becoming physiologically stressed because their vascular supply is down here in the dermis, and so they're having problems taking on oxygen, getting rid of carbon dioxide and other waste products. And when they become physiologically stressed, a gene is turned on that causes them to begin to produce the protein keratin. And a keratin is produced by rough ER, packaged by Golgi apparatus. And so in this layer, we're able to see a granular pattern to the cytoplasm. So we call this the stratum granulosum. Now with models, there are always nuances, and what they try to do is show thick skin over here and thin skin over here. So when we're dealing with thin skin, we'll just ignore this little thin, more transparent layer because it wouldn't really occur in thin skin, although it would occur in thick skin over here. And so the upper layer then is a layer of dead cells that we call the stratum corneum in thick skin. And corneum refers to horn, which is similar to the construction of a horn, is where this material, which is where it gets its name.
Okay.
What they try to do on this end is show you thick skin. And so we would see the same layers we just talked about, the stratum bacilli, this single row of cells, the stratum spinosum, which is where the cells are beginning to flatten out, the stratum corneum, the stratum granulosum, where the cells are producing keratin. And then what we end up with is a layer where the keratin is real pliable, and therefore the cell boundaries are hard to see. And we end up with a thin layer called the stratum lucidum, which is unique to thick skin, and then a thick layer of stratum corneum at the surface. Now, when we look at the dermis of the skin, to keep the epidermis attached to the dermis and to increase that surface area for nutrient exchange between the dermis and the dermis has many upward folds. And so these upward folds are called papilla, little finger-like folds. And they're more obvious in areas where pressure normally occurs and friction, the palms of the hands and soles of the feet. They're less numerous in areas where there's protection from hair, for example, like in the scalp. So when you look at slides, you'll see high amount of folding in thick skin and less folding in thin skin. Because this folding occurs of the dermis, then this folded area of the dermis is called the papillary region of the dermis. And it's comprised of areolar connective tissue, which has a high vascular network to provide nutrition for the epidermis above it. The area below it, all this area here, is the reticular region of the dermis. And the reticular region of the dermis is comprised of dense irregular connective tissue with a lot of special structures that we're going to come back and look at in a few minutes. And then as we get to the hypodermis of the skin is dominated by adipose tissue, which is areolar connective tissue dominated by adipose cells. And this layer is our insulative layer to protect underlying organ systems and to protect us from heat loss. So when we look at the specialized structures within the skin, we have a nervous component investment that we're going to look at that provides sensations. We actually have hairs that provide sensations and protect the underlying epidermis. And then we have glands that produce various products for us. So we're going to start with the glands and then kind of work our way through the sensory apparatus of the skin. So there are two types of glands depicted on this model. This gland, which is a tube that's coiled at the bottom. And if we looked at this in a microscope, we would see that the tube is lined with simple cuboid epithelium all the way up to here. And cuboid cells typically are secretory cells. So this is a sweat gland, what we would call a sudoriferous or eccrine sweat gland. And it's responsible for producing the watery perspiration that allows us to maintain heat by perspiring and dissipating heat. And it also releases things to the skin surface that pheromones and other things, glands similar to this that allow us to unknowingly communicate with each other. Now the other gland we have is this gland that we can see in a surface view, and then over here we can see it cut. This is called a sebaceous gland because the word sebum is a reference to oil. And so sebaceous glands are typically associated with hair and hair follicles, and they secrete oil to help lubricate the hair. So the classic way we would see them in lab on a slide is associated with hair follicles like this. And they secrete a more viscous perspiration that's lubricative in function. Now hair itself originates from the epidermis during embryonic development and migrates downward. So what we can actually see is if we follow this bottom row of cells of the epidermis, they are continuous with that sebaceous gland we just talked about. They actually go all the way down around the hair follicle, back up and around. So hair is really embryologically epidermally derived. And hair is constructed of dead keratinized cells that are being produced by mitosis at the bottom of the hair in what we call the matrix here. And blood supply is important to controlling that mitotic divisions that are occurring here to give rise to the dead cells that make hair grow. So when we put people on chemotherapy, it arrests the mitosis here, the hairs break off at the skin surface, and it looks like their hairs fall out. When there's a gene that causes these capillary beds to close off based upon the presence of testosterone, which explains why men typically bald much earlier than life and to a much greater extent than women do. So if we look at this, then we have a hair, associated with our hair, we have a muscle that connects to the hair root and to the underneath of the epidermis. This muscle contracts to stand hair up. So you've seen it in a cat or a dog that's cold or a cat that you scare, all their hair goes up. It's twofold. It makes them look larger and it creates a dead air space that cushions blows. So when we get cold, we try to create a dead air space to create a better insulative layer. Unfortunately, for most of us, we don't have enough hair to accomplish it. And when the skin is pulled by this muscle, it creates goosebumps around the hair follicle here. That explains why we get goosebumps. So this muscle is called the erector pili muscle. So when we look at the nervous system components here, we actually have modified cells associated with our epidermis here, which are for very fine touch, which we call Merkel's discs. A little lower in that, inside these upward folds of the papillae, we have sensory apparatus for touch that we call Meissner's corpuscles. And then the load of Meissner's corpuscles allows some skin to be much more sensitive than other skin in terms of elaborating the differences between one touch and two touches to the skin surface. And then deep toward the hypodermis, we actually have a laminated structure called a Pacinian corpuscle. And it's built like an onion, so if you cut it on one plane, it looks like a slice of onion. And if you cut it on the other plane, because it's layered, you can see the layers that would make it up. And this responds to pressure, so it's deeper within the skin than the Meissner's corpuscles at the surface.